Before we start our meiosis notes, we're going to do a little bit more on mitosis basics just as a review. So, what moves the chromatids during cell division? Remember, that is your spindle, or your spindle fibers, I should say. What anchors the spindle, or where does it come from? That's the centriole. When does the nuclear membrane dissolve? This is what's around the nucleus. It dissolves toward the end of prophase. And then what structure holds the individual chromatids together? Centromere is the middle of the chromosome. So, quick review then. In mitosis, if a human has 46 chromosomes, how many will each daughter cell have? If you remember, mitosis makes an equal copy. So the daughter cells will have 46 each. So it's very important to remember that the chromosome numbers are the same from beginning to end. So if a dog has 72 chromosomes, how many daughter cells will be produced? Once again, kind of a trick question. No matter what, one cell makes 2 daughter cells. And then how many chromosomes in each? 72 in each daughter cell. All right, so meiosis. Really important to note here. Meiosis is not a cycle. So it starts in interphase and it ends as a zygote. These zygotes do not redivide and make more. Zygote's it. That's the end. So meiosis is a step-by-step process, but it is not a cycle. So meiosis then is cell division which produces germ cells, or another term you'll hear is sex cells. These are things like your sperm and egg cells. At the very end of meiosis, one parent produces 4 daughter cells because there are 2 divisions. So we have something called meiosis 1 and meiosis 2. And the daughter cells have half the number of chromosomes. So in a human, the original cell has 46. At the end of meiosis, each cell has 23. Because remember, you get 23 from mom and 23 from dad. So more chromosomes. First, we need to know what a gamete is. A gamete is another word again here for a sex cell. And human gametes, like I just mentioned, have 23 chromosomes. But humans have 46. And like I just said, you get 23 from the egg or from mom, 23 from the sperm or from dad, and that makes 46 total. And this only happens at fertilization. So a couple new terms here. We have the word haploid and diploid. So a cell with N, which N means the number of chromosomes, so just N number with number of chromosomes. So that's your basic. So 23. So N is always going to be haploid or half. So haploid means half the number of chromosomes. So you have to combine with something to get your full number. Diploid, on the other hand, di, remember, means 2 or double. So diploid is going to be double the haploid number. So 2n, or 2 times this number, which would be 2 times 23, equals 46. And this is something, don't get too confused. We will go over this in class. So a little picture to kind of show it here. You get 23 from mom, 23 from dad, which make you with 46. So other animals. So what are the chromosome numbers of other species? Just to kind of put things in perspective, a fruit fly has 8, a dolphin has 44, a chicken 74, an ant 2, a fern has 630, and then humans, we have 46. And that's a number, by the way, that you need to memorize. So does the complexity, remember complex is how unique or how many different functions you have. So like we said, a human's more complex than like a snail. Does the complexity of the organism have anything to do with chromosome number? The answer here is no. Because if you look, we have 46 and we're pretty in line with dolphins there. But chickens are not as complex as we are, and they have 74. Or you look at a fern, this is a plant, it doesn't move, it doesn't do any of the processes that animals do, which has 630. So just keep that in mind, that no matter how many chromosomes a species has, it doesn't really have anything to do with the complexity. So what else can chromosomes tell us? They can also tell us something called a karyotype. So this is going to be, this is going to be a picture of all the chromosomes of an organism. And this is a human, because we have 2 at every number. And then over here you have X and Y, which, so 22 plus 22 is technically 44, plus you have 2 more here, which gives you your 46. And then what do you notice about sizes and shapes? The largest are up at the top, the smallest are at the bottom. And shape-wise, they're paired with ones that are similar in length or shape or banding patterns. So what does a karyotype even tell us? So first, like I said, look at sizes and patterns. It also tells us gender. So males are XY, females are XX. So males have that Y chromosome, which females don't. And there are some disorders. We'll get into this much more in the genetics unit, but Down syndrome, for example, has extra 21 chromosomes. So if we go back a picture, at 21, there would be a third one drawn here because it didn't separate during meiosis, which we'll get into. So during meiosis, very, very major thing to know here. Make sure you know this. DNA only replicates once, but the nucleus divides twice. So you go from 1 cell to 4 cells. This is how it's different than mitosis. And those terms again, diploid, haploid, the parent cell is diploid or has double. So in a human it has 46, whereas the resulting cells, such as an egg cell or sperm cell, have 23. So why do we even do meiosis? Number one thing here, provides variation. If you look down here, asexual reproduction, they come from a single parent and they are identical to the parent. This means that if something were to ever happen, the population wouldn't survive because everybody's identical. So the fact that we do sexual reproduction means we have a lot more variety in our genes, in our offspring, and the gametes are randomly combining. So another term you need to be familiar with is homologous chromosomes. So they have approximately the same size and banding. So everything you see here is a homologous pair. And you're probably thinking, okay, we learned chromosomes as this. Now to even make it more confusing for you, in a human or in the karyotype, each one of these lines is considered a chromosome. Okay, so each line is a chromosome. That's how you get 46. So 2 divisions, like I said, we have meiosis 1, meiosis 2. So meiosis 1 is the first division. You will notice some stuff is similar, just the way maybe things line up or the way things happen, but there are some major differences from mitosis. And same thing, if I were you, I'd sketch these, pause the video, and sketch them as you go. So interphase, though, is the same as mitosis. The chromatin condenses into chromosomes, and then they're going to replicate. Remember, that's the S phase. So first up, we have prophase 1. Very important when you're taking a quiz on meiosis or on the test. If you don't have that Roman numeral, it's wrong. Because if you just see the word prophase, you know it's mitosis. If you see a 1 or a 2, you know it's meiosis. So same thing as in mitosis, the centrioles start to form the spindle fibers, but big, big, big difference here. The chromosomes find their homologous partner. Homologous is the one that is most similar to them. So this big one here wouldn't pair up with the little blue one because they're not similar in size. It's going to pair up with the other large chromosome. Also, something called crossing over can occur, and we'll talk about that in just a second. So crossing over is another way to add even more diversity or more variety, because what happens is, and I'll demonstrate this more in class, but you have we'll draw them different shapes. You have two of your chromosomes, and they're going to touch and cross over. So then at the end, you would get a chromosome like this, and maybe and then a chromosome like that. So they're going to cross over, and then the genetic information from this one went here, and the one from this one went here. So now those chromosomes are even more different. And then here's just kind of showing it to you a little bit better of a visual. Paternal means father. These are terms you should probably get to know before we get to genetics. Maternal means mother. So this is the father's chromosome and the mother's chromosome. And then notice they switched arms here. And then at the end, here's your end result. So next you have metaphase 1. Once again, the spindle fibers attach to the centromeres, but that's really all that's the same, because now they're lining up in their pairs of chromosomes. They're not lining up individually. So as you can guess, in anaphase 1, when the spindle fibers shorten, which once again is the same as mitosis, they're pulling apart the pairs. So you have a full chromosome here. You don't have the chromatids. And then telophase 1, spindles break down, the chromosomes unravel, same as mitosis once again. But now they have half the number of chromosomes as the original cell, because the original cell had 4, and now each one of these has 2. Next step is interkinesis. So this is the step between meiosis 1, meiosis 2. So you have like interphase, then meiosis 1, then interkinesis, then meiosis 2, then cytokinesis. So the way to remember this is it's basically interphase plus cytokinesis. So if you were to combine these words and make it what happens in the middle, there you go, interkinesis. This is basically just like a giant timeout. It's just between divisions, okay? Nothing happens, no replication. This isn't like normal interphase. It's not named interphase, therefore interphase processes don't happen. So now meiosis 2. This will look much more familiar to you because this is going to look like mitosis. So the spindle begins to reform, the chromatin goes back to the chromosomes, prophase 2. Metaphase 2 looks just like mitosis. They're lining up in the middle. Notice how it's attaching to the centromere here. And then as you can guess, in anaphase 2, the spindles are pulling them apart into chromatids. But I know, once again, very confusing. It's technically called a chromosome. So it's splitting a chromosome into a chromosome. I don't know why they do that and like to confuse us, but just still remember it the way we originally learned it. And then telophase 2, you have 4 daughter cells forming here. They reach the poles, they're starting to pinch in, but you have it happening for 2 cells. And then finally, cytokinesis. So it ends in 4 haploid cells. So for the organism that this was for, our original cell had 4, but each one of these has 2. So 2 chromosomes in each one. So here's your meiosis kind of picture overall. Meiosis 1, you notice there's a little bit of crossing over here where they're swapping colors. Then they divide till finally you have half the number here at the bottom than you did at the top. So another thing we should talk about is eggs and sperm. So meiosis usually results in either egg or sperm cells or pollen and ovules in plants. Pollen is a male. ovule is a female. Okay, what's the difference? Big factor here. Female meiosis, you only get one usable cell. But in males, you get 4. That's why women produce 1 egg a month through their menstruation or their menstrual cycle, whereas males have unlimited quantities of sperm. Because every time a male does meiosis, they get 4 cells, whereas every time a female does, she only gets 1. So another thing to note is that females, when you're born as a female, you have all of the eggs you're ever going to have. You don't produce any more during your lifetime. So you have all the eggs you're ever going to have, and they hang out in interphase. If you remember, interphase, or more specifically G0, is like cell timeout. So all of your eggs as a female hang out in G0 until you hit puberty and start having a monthly cycle. Then you ovulate 1 egg a month. But when you ovulate that egg, that's when it goes through meiosis. Okay, so all of your eggs are in interphase until they divide out to make your usable cell. Now males are different because they don't start even producing this till puberty, but they're born with no sperm. They just produce an unlimited amount of sperm throughout their lifetime. And then a key thing to note here, fertilization is when an egg and sperm combine. So a couple pictures just to show that a little bit better. This oogenesis is a female. Okay? I don't know how you can remember that, but just try to remember that oogenesis is a female. So if you look, here's our little timeline before birth, after birth. So here's all your eggs. And then they go through this after puberty. So if you look over here to this diagram, they start here. Your egg is much larger than your 3 polar bodies. So you get an egg and 3 polar bodies, which basically just disappear. They're not usable. They can't function for the body. They just disappear. So that's like the previous slide said, you get 1 usable gamete. Meiosis in males is called spermatogenesis, which is much easier to remember. Sperm is produced by spermatogenesis. So same thing, except this one, they all have 23 chromosomes. So 1 of these can produce 4 usable gametes. All 4 can be used. So spermatogenesis is much more efficient for a male versus a female. And then just another final step on problems that can happen during meiosis. This goes back to the Down syndrome part. So if chromosomes don't separate, it's called non-disjunction. So a junction is like coming together. Disjunction means not coming together. And non-disjunction means not separating, basically, or not coming together. So in this picture here, you have your 2. They separate. Okay, so we're still on track here. But then look at what happened. These ones separated, but this one didn't. So here's how this can happen. Let's say this is the female egg cell. If that has an extra chromosome and it gets fertilized by a sperm cell, then this is how you would get a child that has Down syndrome, okay? Because you have an extra chromosome. Now, something else, there's some other symptoms that we'll talk about, like Turner syndrome or Klinefelter, but some of them where you have too few chromosomes. That would be if, like, this was your egg and the sperm fertilized that. Then you have 1 less chromosome. And all of this, don't worry, we'll go over way more in class, and some of it we will get into more in the genetics unit at the end of the semester.